Hello there. This is Mr. W with another episode of Mr. W Live. I'm going to adjust so I got okay. Now I think I got this. I think I am live again with all my good friends from around the world who have come here to hear my biology lesson. And that is just a pretty thrilling thing, I've got to say. I mean, who would have ever thought that, I could be teaching a biology lesson with people around the world, like with Jyoti and with Spoutnik EADS. Spoutnik, that is really something. So I'm trying to do a couple of adjustments right here, getting the screen just right. Because I thought I had this just right a moment ago. And technology, it's kind of wondrous. I mean, how could I be doing what I'm doing without this incredible technology? But there we go. Now I think we got this. I'm going to move this over a little bit. That is pretty good. That is what I wanted. I got Quentin. I got Jody from AK. Jody, you got it. Jody from AK. This is fantastic. Jody, that is so exciting. Guys, I don't know if you know this, but Jody is one of the first teachers to ever get a teacher subscription. So Jody, I am just thrilled to have you on here. Oh, Jody, that is okay. Now, let me see who's on here. I think what happened before was that I hit the quit button. I got 12 people here. That's pretty fantastic. I've got Quentin from France. Once again, Quentin, je veux vous dire, ça va, je suis très heureux d'avoir un étudiant français dans ma classe. C'est incroyable para mí. Pour moi, I just switched into Spanish. This is a big problem that I have. And Spoutnik, you're also from France, and I've got Flora from California, that is, and Jyoti, you're from Illinois. That is fantastic. Guys, that is so great. Guys, I am so excited about this show that I did something kind of silly. I put together, I made a song about it. I actually sang that song for you guys last week. But then, I'm really lucky. I live in the Bay Area, and we have a beautiful park that's close to us. And, it's okay to go to the park as long as you take appropriate social distancing protective measures, which is exactly what I did. And I'm going to put on my headphones so that I can hear things a little tangled up. Excuse me for 1 second. Just going to undo this cord now. Oh my goodness, I've got quite a tangle here. It's quite embarrassing. All right, so here we are. Okay, now I'm back. Jyoti, you saw it. That's fantastic. And Quentin, you're telling me that my French is good, and Flora, you're learning French. That's all very exciting. So guys, I thought that I would play this for you, and I know some of you have seen it because, you guys are the best. And here it is. I'm going to say a couple of things because here we go for Mr. W Live. Let's do it. Oh no, that wasn't what I wanted to have happen. Let's do this. Let's do this. Let's do this. Here it comes. Mr. W's Live. That's me. On YouTube. By the beach. Tuesdays and Thursdays, 2 p.m. PST. To help you learn AP biology. This BIOMANIA system will help you to review. We'll show you how to write your FRQs. Ask Mr. W all your questions, he's live. We'll guide you to a 4 or 5. Oh yeah. Yes, guys, here we are at the Mr. W live show. We're going to learn some AP biology. We're going to review for the AP exam. This is so exciting for me to do this. I'm sliding around on my chair, but that's kind of fun too. And here we are, ready to go and crush AP biology. Yeah. All right, so you guys are talking about some other stuff. We've got Matthew here. And holy cow, Jody, I just saw this comment. Are you doing 6 hours a day of biology, Jyoti? Or are you doing 6 hours a day of French? Holy cow. The review varies. Sometimes I read the textbook, FRQs. Good. All right. Well, guys, here's the deal. I just got to say this at this moment. I'm just going to jump over here. And I'm going to go here. And we're going to go to Science Music Videos. And maybe we're not going to go to Science Music Videos. Maybe I'm going to have to open up another window here. Window. Let's go to File. New window. Here's a new window. Here we are. Let's adjust the size of that window. And yeah, there we are. There we are. Let's hit this button for sciencemusicvideos.com. And I just want to say, guys, if you're going to spend hours a day reviewing, what I'm going to encourage you to do is to review by using BIOMANIA or doing other stuff. Like Jyoti, I just totally want to encourage you that if you're going to spend all this time reviewing, it's much more effective to review using my quizzes, flashcards, so on and so forth. So like, for example, you could hit these flashcards over here, and here they go, and I've got 170 of these that are, relevant to this year's exam. So just uncheck 7, 8, keep those science practices, hit begin practice, and you'll just randomly get flashcards from a huge set. Describe how proteins fit into the cell membrane. Who knows with this year's test if that's important to know or not. It's actually impossible to know, but you got to learn a lot of biology. And once you get saturated with that, I want to encourage you to do 2 other things. If you're finding that there's an area where you're not as strong as you'd like to be, so like for example, cellular respiration or mitosis, the cell cycle regulation, then just go here. And you can read through the tutorials, and that's fine. But what's much, much more powerful than reading at this point is to quiz yourself. Just quiz yourself like mad. Because it's all about what's called the testing effect. And what the testing effect is a proven thing in cognitive science. I have completely adopted it as a teacher, is that the more you test, the more you learn. And that's because tests are so great in and of themselves, but tests show you what you do know and what you don't know, all right? So if you test yourself regularly, you're constantly getting a sense of like, don't know that, need to work more on that, and that results in learning. It results in learning much, much more than passive forms of education like just, and Jyoti, don't get this wrong. I mean, I noticed that you said I'm reading the book. Reading the book is fine. But you'll learn much more per hour if you spend your time testing yourself, answering questions like this. Like, just be sure you're following which of the checkpoints below determines whether a cell will progress from G1 to the S phase. And I mean, this is pretty obvious. There's a checkpoint at 1, a checkpoint at 2, a checkpoint at 3. Now let's say you didn't know, and you put in 2. And then what you'll get in my system is a hint. And then you'll think about that. Find the checkpoint that's located before S. Oh, well, it's got to be 1. Now, here's the deal. You're not going to get that checkpoint again for a couple of questions. And that is called the spacing effect. And the spacing effect is another very, very powerful aspect of learning. Because what you've got to do is you've got to give yourself a chance to forget things a little bit. Okay. G1 checkpoint, you guys are right with me. That's fantastic. All right, Jody. I'm right there with you. Jody, you can actually, I wish this were more like a Zoom kind of thing, because Jody, then you could talk about your experience of having your kids using science music videos. And I hope it's been a good experience. I think it has been, because you've continued to use it. And again, we're kind of stuck now. It's not going to be a time when we're going to be able to do a lot of labs. So this is the time to be doing interactive stuff. So anyhow, that was just my little soapbox about interactive learning, and that's because I'm so into interactive learning. So here was our, oops, we already did that. Here's our agenda for the day. So at our last session, people asked to do two things. They asked to do some review about viruses. So I thought we could do a musical review about viruses. And viruses are an incredible disruption to cellular processes. So it's not in any way impossible that we might experience viruses as something that comes up. This is incredible. This is what happens when you don't silence your phone before you do your broadcasts. That's a good lesson that I've learned. Okay, so anyhow, we're going to do a musical biology review. I'm going to put myself over here on the right. There I am. So a musical biology review. We're going to do a cancer biology review from BIOMANIA. The student requested that. I'll open it up to your questions, and then I've got a little biology quiz. And so, Jody, thank you so much. Okay, Jody, I just want to say that, again, the more you quiz yourself, the better you're going to be at applying it. And this is kind of weird, but people talk about holistic learning and they don't think of flashcards, but like doing flashcards is a way to get your brain to interconnect different topics. And again, this is all proven cognitive science. You might want to check out my favorite book about this. It's called Make It Stick. It's not an advertising book. There's an advertising book called Made to Stick. This is Make It Stick. And this is all about learning, memory. I wouldn't say it's really about the brain, but it sort of keys into the way that the brain works. And fantastic, we've got Emily here. And Emily, I'm so glad, because you asked me to do this cancer-related question, cancer cells with the flow chart and stuff like that, and I've got that. Though I'm a little worried. I think I might do that again tomorrow in our class session, but that's the way it's going to be sometimes. So let's see. What do we got here next? That's our agenda for the day. Let's go here. Let's go here. All right, so we're going to do I'm a Virus, and I'm not going to sing it. I'm just going to play it. But here's the deal. People learn in different ways, and the lyrics are going to come up on screen, because that's how I do all my music videos. I put the lyrics right on screen. So what you want to do is do one of two things. You can just watch on screen, or if you want to, what might be better for you is you can do this, and let's go like this, and here we are. And if you go to contact, etc., that's where I got my music videos. And the music video that I'm gonna play right now is I'm a Virus. So here we are, music videos. Just scroll down to I'm a Virus. And if you scroll just below the video, there you can see all the lyrics, okay? So again, you can just watch it on screen. You can read the lyrics. You can read the lyrics and sing. I don't have a karaoke version of this song. I'm gonna work one up. I gotta work with my music producer, Max, to get ready for that. But let's go ahead and do I'm a Virus. So you guys ready? And here's the thing to do. Here's your little task. As you start, as you watch, think about what questions you have about viruses, because, think about the main features of viral life cycles. There's the lytic cycle, the lysogenic cycle. We've got retroviruses, we've got DNA viruses, we've got RNA viruses. SARS is an RNA virus. That's a lytic virus, not a lysogenic virus. And anyhow, think about your questions, and we'll talk about it at the end of this song. All right, you guys ready?
I'm a virus, an infectious particle, an anathuga pirate, the genuine article, full of chicken pox, West Nile, influenza, yellow fever, AIDS, herpes, also coming right at me. I'm not a cell, not an independent organism, don't even have my own metabolism. I only reproduce myself by taking over cells, then I bust them apart, no wonder you don't feel well. The structure is simple, mostly proteins and genes, packaged as a sub-micron killing machine. My capsule's made of capsids and self-assemble. Whenever cells see me, they start to tremble. Inside my capsid is where my genes reside. I got DNA or RNA on the inside, and the rest of me is various proteins. Whose role is assisting in assault cells and taking control.
I'm a virus. I'm a virus.
The Lytic cycle shows one method of attack. I attach to the self-certification like attack. Inject my genes inside, use my victim machinery to make my genes and proteins. Reproduce some more of me. Step 1, tail. 5 is grab the membrane or the wall. 2, inject my genes. The cell is gonna fall. 3, use the victim. Polymerase and ribosomes to synthesize the proteins. Replicate my genome. 4, tail piece and capsid self-assembly. 5, I'm all together now. Look at all my progeny. 6 is lysis. I burst the cell apart. Now I'm looking for more victims I can heal and depart.
I'm a virus. I'm a virus.
Sometimes instead of lysine, I lysogenize, which happens after I inject my genes inside. I slip into your chromosome, deep inside your room. A menacing and silent presence, bringer of doom. Integrated in you, your DNA's my home. And see what happens when you replicate your chromosomes. Each copy that you copy copies me as well as you. You might make a million copies, there's nothing you can do. My lurk and hide genes call it Provage or Papyrus. And if you got another name, just call us a virus. We'll write it on papyrus till the moment I emerge from your DNA and lytic. Now it's self-destroying surge. And suddenly your cells are dying. Watch me as I lies them. I'm a pirate, so forget you plan the program now denies them. We cells are virus factories and making millions more of me. I'm losing cells and now I'm free. Infecting is my destiny. Now meet my associate, the virus HIV, whose helpless target is a cell called T. HIV causes immunodeficiency, raising cancer and infection vulnerability. Remember that the T is an essential cell in the immune system army, it's the general. So knocking out the T cells sets the stage for cancer germs and viruses like me to invade. HIV's genes are made of RNA, outside its capsids, a phospholipid membrane, the skin stolen from the cell blast dispatch, studded with proteins that landed attack. Attached to a protein on the T cell, CD4, inducing that cell to open up its door, inviting in HIV's enzymes and core. The T cells were already done for. HIV is a retrovirus, its sex routine has to do with transforming its RNA genes. Reverse transcriptase takes its RNA and reverse transcribes it into DNA. Then this DNA integrates into your chromosomes like a lysogenic protein in your genome. This proviral DNA is transcribed and translated as new virus genes and proteins get created. And like any virus, all those particles self-assemble into new viruses, which of course resemble the original particle that started the infection, provided by sex or a needle injection.
Wow, was that fun. Guys, I know that there's a certain asynchronous nature to this, but now would be a good time for you to shoot me any questions that you might have about viruses, lytic cycles, lysogenic cycles, retroviruses, DNA viruses, RNA viruses, and anything you got. One thing that I do want to point out is that on sciencemusicvideos.com, if you go here, AP College Bio, if you go into the section about gene expression and regulation, that's where you'll find a tutorial about ribosomes. Excuse me, what in the world was I thinking? If you go into topic 6, gene expression and regulation, that is where you'll find a tutorial about viruses. Here it is. And, you know, it gives you a basic introduction. Don't worry about this PowerPoint. It's embedded in, you know, the environment that I've created this website in, which is called WordPress. For some schools, that winds up getting blocked, and if you have a school email account or web address or something like that, it won't show up for you, and there's nothing we can do about that. Nothing we can do. But anyhow, it'll take you through the lytic cycle, and it'll take you through the lysogenic cycle, and it'll take you through the life cycle of HIV. You know, I haven't yet developed specific material about SARS-CoV-2, but one day I will. So let's see. And let's see, I got some questions. Daniel Chen, if I don't have an AP Bio teacher, can I still use the flashcards and test? Daniel, you can, but you have to subscribe, and I'm just going to take a minute and show you. So Daniel, what you would do is you would go up here, and you would hit, well, there's a bunch of ways you can do it. You see over here where it says get a student subscription in the sidebar? So you do that, and you fill this out, and Daniel, it's $19. It's the best value in biology education. You'll learn so much, so I just want to encourage you. Okay, so now let's go back to viruses. I'm going to go here, and we'll bring up viruses again. Here we are. And viruses. Okay, so GOT. So the lytic cycle is an attack and destroy cycle. So essentially, the virus, which is shown here, lands on the surface of a cell. Now, this is a kind of virus that's called a bacteriophage. You've undoubtedly seen pictures of the SARS-CoV-2 virus. It has a different shape, but basically, you see these little legs over here? Those are molecules that have some affinity for surface molecules on whatever cell the virus has become specialized to attack. So like, for example, just to talk about the thing that's on everybody's mind, SARS-CoV-2 has surface molecules that are specialized for attaching to a surface molecule on the epithelial cells. Those are the lining cells of our lung tissue. And there's a kind of lock and key fit, and of course this isn't like enzyme and substrate where it evolved for our benefit. This is something that the virus evolved for its benefit and for our detriment. Anyhow, the docking of that surface molecule on the surface of the SARS-CoV-2 virus with our epithelial cells induces some changes in the membrane properties that cause that virus to basically enter into the cell. You know, that's not a natural thing. That's a ridiculous word. Of course, it's the most natural thing in the world, but it's certainly not an adaptive thing for us to let viruses in. And, you know, cells have selectively permeable membranes, and those membranes keep out bad stuff and only let in good stuff. But in this case, the virus has evolved a mechanism to undermine that. Okay, so back to these bacteriophage. Basically, in the case of these bacteriophage, they're kind of injecting their genetic material inside. That genetic material might be DNA, it might be RNA. In the case of these bacteriophages, my guess is that it's mostly DNA. Well, that's sort of what's represented here, but you know, it could be RNA just as well. SARS is an RNA virus. Now, what's not really shown here in detail is this. The injection of that viral material leads to protein synthesis and DNA replication using the cell's own machinery. So like, for example, in the case of COVID-19, well, first of all, I hope it's not yours, but let's just use yours as an example. If you were infected, your cells, ribosomes, would synthesize viral proteins. Your DNA polymerases, or in this case RNA polymerases, would synthesize more viral RNA. So the cells are hijacked. They're taken over by the virus, which converts the cells into virus factories. That causes a tremendous, tremendous amount of cellular damage. And that is actually what's causing the health impacts of COVID-19. In other words, it's for some people so severe that they wind up being put on respirators. Those respirators are themselves a traumatic experience. I hope that no one here has experienced the virus directly in terms of them being ill themselves or having people in their close family, their loved ones, experience it as well, because it seems like it's just a terribly difficult thing if you get a bad case of it. But in any case, that's the lytic cycle. At the end of the cycle, the cell winds up getting lysed. It winds up getting broken open. Now, that's very different from the lysogenic cycle. That's what I'm showing in this diagram over here. And the lysogenic cycle has an added complexity. So for the lysogenic cycle, we start over here. The virus injects its genetic material, and that genetic material incorporates itself into the host cell chromosome. So that's what's happening here. That F is called a prophage. It's incorporated genetic material. Now, every time the cell replicates, and if these are bacterial cells, they replicate very quickly, but even your cells, for example, not a red blood cell, a white blood cell, it'll replicate every couple of hours. It doesn't matter. It's an exponential growth rate. So over time, you'll wind up increasing the number of cells that are infected with the virus. Now, at some point, some trigger, and I can't tell you what that trigger is, and I don't know if anybody can. Like, it's certainly at the limits of my knowledge, maybe other people know, is going to induce this embedded phage. Now, imagine millions of cells like this. They all have these embedded phages. They're called prophages if it's bacterial. They're called proviruses if it's a eukaryotic cell. They'll break out, and then they'll sort of finish the lytic cycle. But again, instead of one cell finishing this, it's millions of cells finishing this process. Okay, so there's a question about Melissa, who came here to say great work, and thank you, Melissa. So for lysogenic cycle, the DNA is actually incorporated or integrated. That's exactly right. And someone is saying information on viruses weren't going to be on the AP exam. And of course, let's just look at this, okay? So here's our AP biology curriculum. Unit 1, chemistry of life. Unit 2, cells and membranes, cellular energetics, cellular respiration, photosynthesis, cell communication in the cell cycle. Heredity is mitosis, meiosis, genetics, dihybrid crosses, linkage, recombination, the whole shebang. And then Unit 6 is gene expression and regulation. I think that viruses is squarely within this topic over here. In other words, viruses have genes, and how do those genes get expressed? Through the lytic or the lysogenic cycles. I've certainly taught my students viruses, but no one knows what's going to be on this test. But I can say this: viruses, they're really not in here in natural selection evolution. Though of course, the tragedy of this year is that natural selection and evolution, that's the most important unit in the course. So the fact that they dropped that, it's tragic. But I'm sure the College Board really thought about this hard. And ecology is also incredibly important. That's not going to be on the test either. Viruses, of course, they impact ecology. There's a way in which viruses act through what's called density-dependent regulation. The higher the density of a population, the easier it is for a virus to jump from one host to the next. So there are certainly ecological connections to be made between viruses and ecology, and there are evolutionary interactions everywhere. But I would say this, I'm not sure what your name is, but I would say I would study viruses. And I think that if you do this, if you go here, and you just hit this one unit that I have about viruses, you're going to be fine. You're really going to be fine. And, I think this is all you need to know. Viruses are hugely important in the history of life. To leave a biology class without understanding viruses would be a challenge, especially in this era of COVID-19. Okay, so any other questions? Flora, you're asking for a eukaryotic gene regulation. I think I got a list. I'm going to leave that for the next, but I'm going to make a note. Eukaryotic gene regulation. Gene regulation, and we'll put that down there. But Flora, between now and the next time we meet, I just want to show you something. What I want to encourage you to do is go here. Okay, go down here. And this whole topic over here, eukaryotic gene regulation and expression, this is tough stuff. But I would definitely go through this and this. This might be one of the hardest units in the whole course. It's very abstract, it's difficult, but do that. There's another associated unit, and it's about development, animal development. It's mind-blowingly cool stuff. It's one of my favorite things in biology. And there's some important things to know in there too. So, we've got time. We've got, we've got 20 days between now and the AP Biology exam. It'll take you a couple of hours to go through these two topics about eukaryotic gene regulation and expression. So Flora and everybody else, I would strongly, strongly suggest that you go ahead and you do that. And again, if your teacher hasn't signed on for an account, then you do have to get it individually as a student. And it's $19, it's not a big deal. Ask your parents. I'm hoping they'll support you because it's really important. And Emily, I got that eukaryotic gene regulation circled. You just heard my old thing. And chromatin structure, Flora, I'm glad you got that because that's pretty important. Though again, this is such a weird test. Like if this were the normal multiple choice test, there would definitely be some question about eukaryotic gene regulation. What they're going to do with these two FRQs, I don't really know. Experimental design and a biological system that gets disrupted. So speaking of which, let's talk about a biological system that gets disrupted. Doesn't seem to be any more questions. Oh, just want to tell you guys, special deal running right now, $6.99 for access to the BIOMANIA app. You download the thing for free, but then you unlock the content with an in-app purchase. And, if you're my student, it's not a problem. But if you're not my student, and even if you're anybody else's student, you still need it. It's no different from this stuff here. In other words, that BIOMANIA is the same as this BIOMANIA, but a lot of people like the convenience of having the material on their phone. And if that's you, then I would totally encourage you to go ahead and get the app because like $6.99, again, I understand like people are out of work, the tough situations, but like $6.99 is not a lot of money. So, there's that. And then here's another special offer. I'll get out of the way so you can see this. I don't really know how to do this very well. Maybe I'll go here. Yes, perfect. Okay, so here's the thing. I couldn't really do this with a code or anything like this, but if you shoot me an email and just say, hey, it's me, blah, 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 I was watching the show and you talked about a discounted price. I'm not giving away the thing for free by any stretch of the imagination, but I will sell it to you at a discounted price. Just put access in the subject line and then I'll offer that, maybe like a 25% discount. The whole thing's only $19.99 to start with, but I totally appreciate the fact that people are here watching the show. Very numbers. We got 13 people watching the show now. That's fantastic. That makes me really happy. So here we are. I'm looking at the wrong thing. I should be looking at this. Good. Okay, so I've talked about my special offer, and let's go on. So guys, we're going to do a practice FRQ. I hope everybody's ready to do some
AP Bio Review FRQ.
Sometimes when I get the pandemic blues and I start to feel a little sad, I just pull up this file made by the amazing Max Cowan, who does all my music. He also did the Mr. W's live. He did the background track. In fact, he took my voice and he auto-tuned it to make it sound as good as it sounds, because my live singing is, only okay. But I just got to do this again because it just brings a little joy to me, and we need joy. So here's a little joy. Everybody take some in.
AP Bio Review FRQ.
Okay, enough of that. I'm wasting time. Okay, so here's the deal. We're going to do this practice FRQ. I understand that it's very hard sometimes to read on screen. So I put this together for you. So here, sciencemusicvideos.com, right? So here we are. So just here you're at the top, right? Wherever you are, you got this menu. Hit live. Okay, and then you see it says next live performance. Well, that, if I hit this, we would see me. It would be one of these weird recursive loops where you would see me, seeing me, talking about me. It's just too weird. So anyhow, I just put the entire text for the question. I didn't put the answer here, but just note that if it's better for you to go here to Mr. W Live, QOD, then you can see this here as opposed to seeing it all on screen as I show it through the PowerPoint, because that's a hard way to do something, okay? So let's do this. Hey, and you know what would be a great thing to do right now? I know that we've got 16 people on, and that's beautiful. I'm going to take a sip of tea. And while I'd like to do this, I want everybody to do the thing that you're not supposed to do in school. I want everybody to take their phone out, and I want everybody to text. You're all part of these group chats, these big group chats and things like that. And what I want you to do is I want you to send a big group chat out. Maybe you have a Slack channel with your friends. Maybe there's a Facebook group or Instagram or TikTok. I've actually put something on TikTok, though I don't really get it. I don't get it. Anyhow, but the point is I'd like you to invite one person to come on here. And in fact, I think I've said this before. If we get 100 people watching this, I'll sing glycolysis. I'll do a live version of glycolysis. But again, do that thing. Invite people to join us. But in the meanwhile, let's go. Take a look at this diagram, okay? And I'm just going to read it out loud. The image below, well, it's not below, it's on the right, is a schematic diagram of a signal transduction pathway within a cell. Arrows represent activation, while a T, which may be upside down, see the one between JN1 and BCL2, see that's like an upside down T, represents inhibition. So this is, if this were the nervous system, we'd say this was excitatory, stimulatory, and this is inhibitory. Let's just use those words. Inhibitory and stimulatory. And just think stop, okay, or go. So the arrow is like go, and that T is like, oh, my hand disappeared. Stop, like that. Okay, so the overall result of this pathway is apoptosis. One of my favorite words in biology, one of the most widely mispronounced words. Apoptosis is programmed cell death, okay? So, oh, wow. I'm looking about this, a Snapchat AP Bio group. Oh, fantastic. Emily, I'm really indebted to you. Thank you so much. Okay, guys, so we're going to go on. Everybody, I read this aloud. You can read it online. Let's go like this. Oops. Okay, now, this is a description of some of the events that occur within this pathway. I'm just going to read it aloud. Some of the events that occur within this pathway are as follows. 1, tumor necrosis factor, TNF, that's this, it's a ligand. It's a local regulator. It's not really a hormone, but it's a local thing. Binds to and activates specific receptor, tumor cell necrosis factor receptor, TNFR. The activated TNFR activates JN1. The activated JN1 activates caspases by inducing mitochondria to release cytochrome C. So JN1 sends a signal to the mitochondria. The mitochondria release cytochrome C, and JN1 also inhibits BCL2. BCL2, when active, promotes cell survival by inhibiting caspases, right? See, that's an inhibitory link. This is stimulating this, and this is causing these caspases to induce cell death and nuclear DNA damage. Now, you might be thinking, wow, that's weird. Cytochrome C, that's like a central molecule in the electron transport chain, and it's the same one. It's the same one, and the export of cytochrome C from the mitochondria to the cytoplasm is basically a signal to induce cell death. There is some deep evolutionary biology there. The guy to read, and I think some of you guys might be at this level, like you are total biology aficionados. Some people who are going to get negative might say biology nerds, but like I'm a total nerd, and I'm a biology aficionado. I love this stuff. I could do this stuff all day. I listen to books about biochemistry while I ride my bike. So anyhow, Nick Lane talks about that evolutionary relationship, but that is not our issue now. Our issue now is this. Okay? You got a question. Several elements of the system have been implicated in the development of cancer. Some of these are loss of function mutations, all right? Loss of function mutation. Don't be intimidated by that. It just means that the thing, whatever it is, stops working, loses its function, stops working, okay? Based on the signaling pathway shown, describe the effect of a loss of function mutation of each of the following upon apoptosis compared with wild-type cells lacking this mutation. So the three are TNFR, that's this receptor over here, BCL2, that's this molecule over here, and caspases. So in the chat, use this formula to respond. If blank, one of these three lost its function, then the effect on apoptosis would be blank. So I just need something to respond to. You don't have to do all three, but do one of them. And if your birthday is in the first third of the year, which is like between January, February, March, April. Okay, January to April. If your birthday is January to April, respond to TNFR. If it's May to August, then do BCL2. And if you're at the end of the year, September to December, do caspases. I want to see one sentence in the chat. You know, we got, I don't know, how many of you do we have? It doesn't matter, but let's say that we've got 14 of you. So 3 sentences each. You don't have to do it all. It'll make things go a little bit quicker. Let's make it happen. Sentences in the chat. Let's go. I'm waiting. Oh, you need to see this. Sorry. There you go. Okay, so let's see some sentences in the chat while I have a cup of tea. I don't know if you guys know about this, but this is my Science Music Videos mug. I love it. You can get one. Okay. Okay. I got something from Giotti. Let's get a couple of more things, guys. Let's go. Emily. Oh, Emily. Very cool. You use the word bleb. Okay. Okay. Let's go through this, okay? So, you guys are awesome. Let's do this. Okay, so here are the answers. Okay, why don't you guys, I'll read this loud. If TNFR lost its activity, if this receptor over here lost its activity, then the cell would respond less to the tumor necrosis factor signal. right? Cell death would decrease. Apoptosis would decrease because this whole pathway, JNN1 to mitochondria to cytochrome C, would be blocked. So this whole thing wouldn't happen. There'd be less cell death. You wouldn't have caspases getting activated. You wouldn't have cell death. And Emily's saying that bleb. What cells do is apoptosis is actually a very organized, stately process. It's not like the cell just goes, blah. The cell just sort of like, slowly sort of packages itself up and releases these little bubbles of membrane enclosed cytoplasm that are called blebs, and then immune system cells come and gobble them up and they get recycled. It's a beautiful process. And without apoptosis, I tell my students this all the time, this is the result of apoptosis. During development, apoptosis sort of gets these cells that were over here to die. It's sort of a sculptural process of removing excess material. So that's TNFR. Let's now look at BCL2. If BCL2 lost its activity, cell death would increase because the inhibitory effect of BCL2 would be removed, allowing caspases to cause nuclear damage, leading to more cell death, okay? So BCL2, a loss of function, would lead to more cell death, more apoptosis. So less apoptosis, more apoptosis. This one goes, less apoptosis. This one goes, more apoptosis. Let's look at the last one. Okay, if caspases lost their function, then cell deaths would decrease because the caspases cause cell death. Now, here's the big deal. Let's just go wide a little bit. It is so likely that on May 18th, you're going to have some complex visual representation like this that you'll have to analyze. And so I want you to use a combination of the ones that I have on my website, where I have a lot of them, the ones I have on BIOMANIA, where I still have a lot, but not quite as many, and just to really, really get comfortable with sort of looking at these interactions. And I just want to show you one thing. So like, for example, I just went over this with my students, and it's in the regulation unit, feedback, topic 4-5. So if you go into this, and if you go into like blood glucose regulation, I'm not getting a little cold here, you'll see complicated diagrams like this. And one thing that my website is really good at is getting you to work through complicated diagrams like this and understanding the interrelationships here and like what stimulates what and what goes where. I think that's going to be a big factor in your success on May 18th. All right, let's go on. Okay, now, here's the second part of the question. Gain of function mutations are mutations where a cell component gains a capability that it previously lacked. Based on the schematic, on this diagram, identify the components of the signaling pathway that might promote cancer cell growth if they had a gain of function mutation that increased that component's activity. So, excuse me for sniffling so much. In the chat, use this formula to respond. If blank gained function, then cancer would increase because. Okay? So think of something that if it increased its function, there'd be more cancer. Not less cancer, but more cancer. More. Yeah, I'm not going to say more because that would give it away. That's something that teachers do all the time. What teachers need to do more of sometimes is sit back and let their students do more of the thinking. Let's see it in the chat. Okay, let's see a little bit more. There's really only, okay. So, Jyoti, justify. Say why. Predict and justify. You always have to justify. And that because is your justification. Emily, you too. Justification. Why? Want to see a justification? Anybody? A justification? BCL2 is the answer, but why? All right, there we go. Okay, Flora's got it, guys. Everybody look in the chat about what Flora had there. Okay, so here's the answer. If a mutation caused BCL2 to increase its activity, a gain of function mutation, it would block caspases. Flora, you said that, it was beautiful. Less cell death means higher survival rates for cancer cells, increasing cell growth, cancer cell growth. Guys, brilliant. You know what, guys? It's 2:51. We're going to skip the last part. Guys, questions for next session. Next session is Thursday. I've already got eukaryotic gene regulation on the list. I can give a brief overview of that. Oh, there's my neighbor who's got a big motorcycle. But that's not too bad now. So guys, anything that you want me to cover, any topics? There were some that showed up in the chat before. Eukaryotic gene regulation. All right, guys. I want to make this student-centered as possible, so please let me know what you got going on, what you want me to do. I've got eukaryotic gene regulation on the list. Is there anything else? Anything else besides eukaryotic gene regulation? Photosynthesis. I can talk a little bit about photosynthesis. Question. Animal homeostasis, exotherms versus endotherms. Okay, I've got this. And Rhea. Okay, photosynthesis. Light reactions or Calvin cycle? You know what? I have a video. I just want to show you guys. I've got a good, I'll show you something if you're interested in photosynthesis. So let me show you this first. So guys, here, go to AP College Bio, go to photosynthesis. I've got a very good tutorial about photosynthesis. So let's see, Rhea, I want you to go through photosynthesis, and maybe if you still have questions, shoot me an email, because I've really got to cover it. And also, I've got a couple of really good videos about, where is it? Here. So I've got a whole video that explains the light reactions in a lot of detail. So my preference would be to do the Calvin cycle. But I have a question. You know, I did a when I was just experimenting with these, of course I've only been doing it for 2 weeks, I did a lesson on the Calvin cycle. Were any of you guys there already? Like, did you get the Calvin cycle already? Because if you already got it, all right, looks like, okay, Calvin cycle. Calvin cycle's on the list. All right, guys, we've got too much for 1 hour again. So let's see what else we're going to do today. Got some questions. Guys, let's do a couple of quiz questions. Just some random quiz questions. What I want you to do is I want you to have your fingers on the keyboard. Let's see who gets it first. All right, everybody ready? Fingers on the keyboard, ready for this first question, because it is quiz time. It's not exactly AP Bio. It is AP Bio Review, but it's quiz time AP Bio Review. Bio quiz, and let's go through. And Emily, no, it's not. I took it down because it was pretty bad, so I didn't feel good about my first effort. So I'll do Calvin cycle. I'm going to do Calvin cycle, even though I taught you, Emily, the Calvin cycle already. But I'm going to do it again. Guys, quiz time. First question. You guys ready? All right, here's the first question. In some blank, temperature determines sex during embryonic development. think of a type of organism. Let's see an answer in the chat. Anybody else? I don't want to jump up and down yet. All right, you guys got it. Fantastic. All right. Next question. An example of this kind of inheritance in animals is inheritance of mitochondrial DNA, which is passed on exclusively through the egg. What is it? Oh my goodness. Remember, sneezing is not really a symptom of COVID-19. Jyoti, you might not be wrong about fishes because there's an AP Bio question that talks about a certain kind of fish in which they have like a ZW system and temperature can actually change that. So you might not be wrong. Okay, guys. Mitochondrial DNA isn't really the same as sex linked. I think what you're thinking is that mitochondria are only inherited through the egg, but there's a different word for this. There's a different word. This is called, where's my mouse? Non-nuclear, extranuclear, extrachromosomal inheritance, okay? And essentially in animals, in all eukaryotes, you can get genes that are passed on through the mitochondria. In plants, you can also have genes that are passed on through chloroplasts. And that's a different mode of inheritance than what you'd experience in almost like all the other genetics that you might learn about. So just remember that that's an example. Non-nuclear inheritance. Let's look at something else. Another question. Oh, extranuclear. All right. Whoa! Oh, I ruined that. All right, we're going to go on. Answer to this one is obviously oxidative. And this is really cool. So if you don't make ATP, but you still have electrons flowing like crazy, well, when electrons flow down a wire, that generates heat. That's essentially how a toaster works. So there are organisms that can generate heat by decoupling electron transport from oxidative phosphorylation. So you generate heat. It's a way to keep yourself warm in a cold stress environment, especially if you're a mammal. Let's look at this one. In response to lowered body temperature, humans will shiver. These muscle contractions raise body temperature back to the set point. This is an example of what kind of feedback? And of course, you know, the idea is that you stop shivering once your body gets back to the set point. What kind of feedback is that? Let's see some more answers come in. Nice. Nice, 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 nice. Okay, let's do one more because we are at 3 o'clock. Oh, I just taught you this. Well, you should know this word based on what I taught you. In this viral life cycle, a single virus attached to the host cell and then injects its genetic material inside the virus, uses the cell systems for DNA replication and protein synthesis to synthesize new viral components which self-assemble into new viruses. Blank of the cell releases the progeny. Progeny is the offspring. Progeny. You are your, yes, that's right. are your parents progeny? Yes. Got an answer to this question about viruses? Let me see one come in. Emily, I think you're misunderstanding what I mean here. Lysis is the answer. Guys, that's going to bring our work to an end for the day. Okay, so we're not going to do any more FRQs, but I just want to say how proud of you I am. You're coming here. You're working through your biology in the midst of the great pandemic of 2020. You guys are doing it. And I think that with your determination, you've got a hell of a good chance of getting to your goal over here. All right? So guys, stick with it. Keep practicing. Use the College Board questions. Use my BIOMANIA website. Use my BIOMANIA app. Remember the special, you just send an email to me saying access, and I'll give you a reduced price on access to my entire website, $6.99 for access to the app. You guys are the best. I'm going to be here again on Thursday, and I want you to do me a favor. Publicize this. Okay, so just invite your friends. Say there's a really cool thing going on. Mr. W's doing some AP Bio review. I hope you're finding these review sessions useful. And if you are, then just sort of spread the wealth, share the love, get more people to come in to these sessions. The more people that come in, the better. I mean, I'm happy to spend this time with you, and I want to amplify my impact and help more of the thousands of kids who are really in a tough situation in the, COVID-19 shutdown, the school shuttering that we're all experiencing. So, guys, stay well, practice social distancing. I want each of you to invite 5 friends to the next one of these. Stay safe, stay healthy. I'll be here Thursday. Mr. W, signing off. You guys are the best. Take it easy, and I'll see you on Thursday. Let's go right here, and here, and here, and bye, everybody.